Welcome everyone to our reading group. And let me introduce Dominik. So he's a PhD student in Fabian Thesis lab at Helmholtz Munich. He's working at the intersections of machine learning and single-cell genomics. So during his PhD, he spent time in Marco Couturi's group at Apple in Paris as well. He holds a master's degree in statistical science from the University of Oxford and did his bachelor's degree in mathematics at ETH Zurich and Imperial College London. So welcome, Dominik, and he will introduce us this paper called Mapping Cells Through Time and Space with Moscot. Hope I pronounced it correctly. And this paper is just published at Nature. And congratulations. Okay, it's all yours, Dominik.
Thanks a lot for the kind introduction, and thanks everyone for joining this talk. So as outlined in the introduction, I will mainly focus on our recent paper on Moscot. In the end, I'll spend a few minutes on a little bit of outlook or follow-up work of Moscot, which goes more into the direction of generative machine learning. But yeah, let's first do a deep dive to Moscot. And I don't know, how do we handle it now with respect to questions? Do we pose them at any time, or do we collect them till the end? What is your preference?
I think it's okay. Like you can choose. Yeah, like everyone. If you're okay with everyone interrupting you in the middle of the talk, it's okay. If you prefer them to speak at the end of the talk, it's also okay. So we have like 1 hour, and if you don't have other steps, we can also extend this. So I think everyone is free to come and free to go.
But we usually we have like this sort of collegiate, like if it were a coffee chat in a sense, and just have this sort of space where you can query the presenter a bit and figure out why the method worked, why it didn't. So that's an energy that's very much welcome if you would like that.
Yeah, that's absolutely fine. So interrupt me whenever you would like to. So I'm happy with that.
Great, let's do that.
Cool, amazing. Let's kick it off. So in single-cell genomics, distributions of cells often have to be realigned or matched. In other words, distribution of cells have to be linked. This is mainly due to two reasons. First, sequence technologies are destructive, which means you can sequence one cell only once. For example, in the context of perturbation modeling, you cannot measure the same cell under control and perturbation. Or in the context of development, you can only get the measurement at one time point. You cannot measure it twice. Hence, to study and understand which cell evolves into which cell, you have to somehow link two distributions of cells. The second reason for the need of realignment or alignment of distributions is the number of modalities which can be measured simultaneously. So we know there's almost an infinite number of modalities which you can measure in a cell, but the technologies from a biotechnological point of view, they don't allow us to capture all of them simultaneously. But in order to get a comprehensive view of a cell, we would like to measure or bring together as many modalities as possible. And therefore, we also have to align modalities. Now, there are different ways of aligning two sets of cells, or in more abstract terms, there are different ways to connect two point clouds. But optimal transport is maybe the one which is most natural, while not assuming any parametric formulation of the connection, for example, in terms of single-cell genomics, some mechanistically informed coupling. So optimal transport purely works on 2-point clouds and is based on the geometry of those. But let's have a look at the task of connecting 2-point clouds. So in red, we see what we call a source distribution, and in blue, what we call a target distribution, and we want to connect the source distribution to the target distribution. Again, in the context of single-cell genomics, this could be, for example, the source distribution could be measurements at an earlier time point of a developing system, and the target distribution could correspond to a later time point of a developing system. but there are many more examples of that. So we could just randomly link them. Here we see basically some random arrows, now a different configuration, and we could move on like that forever to the exact 720 times. And this is just given that relatively small set of data points or cells, which is 6. And if you think about like upper scale single cell data sets, obviously you don't want to try all of these combinations to get one meaningful one. So the question is, how do we choose the best coupling? And this is where optimal transport comes in handy. So I'm trying to briefly explain the concept to give you an intuition without going too deep into the math. But we need to quantify some notion of cost. I think here intuitively, this might be the Euclidean cost. So we just have to compute a cost function, or I have to evaluate a cost function which measures the distance between a data point in the source distribution and a data point in the target distribution. So we just enumerate all of these data points and all of these pairwise distances, hence we get this matrix. And in the end, we want to find the optimal match or optimal transport match, which minimizes the total displacement cost. So in that sense, this means we just add up the length of all of these arrows and want to find this permutation, which minimizes the sum of the length of the arrows. And in the end, what we get out is what we call a coupling matrix, or in that case, a permutation matrix, which gives us, for every single data point in the source distribution, a match in the target distribution. Now, in practice, we solve an entropy regularized version of this optimal transport problem, which encourages fuzziness. So we won't get this deterministic map, but rather a stochastic map. This has actually a few reasons. The first one is very obvious. If you have a different number of data points in the source distribution than the number of data points in the target distribution, you just cannot find a permutation. Hence, you might want to split mass or allow some data point to receive mass from multiple data points. So there's like this generalization, which is called Kantorovich relaxation, that allows you to split the mass, and hence you get a stochastic mapping. Now, at the same time, we want to add this entropy of the transport matrix because it allows us to compute or to turn this problem into a convex problem. So from a computational perspective, it's just much easier to solve because, it renders it epsilon convex, this problem. And hence what we get in the end is a coupling matrix, which stochastically links cells from the source distribution to the target distribution. And this epsilon determines how stochastic it is. So basically, if epsilon goes to infinity, we get the outer coupling. So every cell will be mapped to every other cell with the same probability because we want to encourage as much entropy as possible. Whereas if epsilon goes towards 0, we will slowly move towards what is also called the Monge map setting, so to the deterministic mapping. But this is computationally more and more difficult. So also if you want to solve a real world problem basically with moscot, you would have to choose that epsilon hyperparameter, and you will realize whenever epsilon is larger, the algorithm will converge much faster than when it's small. At the same time, you don't want to have a completely fuzzy coupling because, you want to still maintain the signal. So this is a little bit of a trade-off of how fast it is to solve versus how directed or how deterministic the mapping is. Now, this is to give a first idea or introduction to optimal transport and why this might be useful. In general, the concept of optimal transport has been studied actually since the 18th century. And then with Marco Couturi's Synchron algorithm or applied to optimal transport in 2013, this became more present in the data science and machine learning community, this concept of optimal transport. However, there were a few limitations of already developed optimal transport applications in single-cell genomics. So first of all, most or all of these applications have been limited to unimodal data. So the nice thing about optimal transport is that you don't have to have any assumptions on the data modality because it's purely based on the geometry of the data. But now with more expressive biotechnologies, for example, technologies capturing multiple modalities at the same time, I will show you an example later, we would obviously also like to study whether we can improve the performance or like improve the meaningfulness of the couplings with multiple modalities involved in the coupling computation. Second, and very crucially, optimal transport in its original formulation is not scalable. What does that mean? So this optimal transport formulation, which I just introduced, which is also called linear optimal transport. Later, we will also talk about something which is called chromoplasticine, fused chromoplasticine, or quadratic optimal transport. So the linear optimal transport by itself is already, in its original formulation, quadratic in time and memory, which prevents its usage to large single-cell data sets. The other type of optimal transport, which we're going to talk later about, is even cubic, which makes you run into memory and also time complexity issues very, very fast. And third, there are these applications, especially Waddington OT were the most famous one. I think the first well-known application of optimal transport in single-cell genomics, published by Geoffrey Schielinger in 2019 in Cell. There are those frameworks, but they are very different in their guidelines of implementation. It doesn't really follow a joint setup and a joint user interface. At the same time, all of these algorithms work very similarly in the back end, and hence we wanted to provide a unified user experience to biologists such that it's easier to access this maybe a little bit theoretical concept of optimal transport also to biologists who might not be too familiar with all the details in optimal transport theory. So we introduced and developed moscot, which we developed such that it can be straightforwardly applied to multimodal data, where we have, for example, multi-omics, site-seq, or also spatial transcriptomics applications. We make it scalable with different strategies. So first of all, we provide GPU acceleration for all algorithms, for all underlying algorithms, and hence it's just much faster to solve. At the same time, we also have a few tweaks on the implementation by building upon OTT JAX, a generic optimal transport library implemented in JAX developed by Marco Couturi at Apple. And the one nice idea, for example, is that you can reduce the computational complexity of the linear optimal transport problem from quadratic to linear whenever you have a separable cost, because you don't have to instantiate, you don't have to really compute all of these pairwise distances simultaneously, but you can actually do it in an online fashion. So hence, we already get, just by a tweak of implementation, to much larger scalability. And second, we leverage recent low-rank approximations of optimal transport, also mainly spearheaded by Marco Couturi in this lab, and show that they are also meaningful in single-cell genomics. And this way, we get to linear time and memory complexity across all applications, even to the originally cubic time complexity of chromoplasticine and fused chromoplasticine problems.
Quick question.
And finally, is there a question?
Yeah, just about not the acceleration, but when going from quadratic to linear. Are you talking about the factored OT or the low rank OT?
Area? This is low rank OT, correct.
Yeah. And that was actually a question I was going to have for another reason, but because it's relevant here. So you think like these hidden factors are relevant study per se? Because I've read Moscot a while ago when it was still a preprint. And I think I remembered mentioning factored OT, but yeah, there's like a lot that could be done there, right? Or even the low rank OT, where you essentially learn bottlenecks, right? So
correct, yeah, that's true. And if your question alludes to like how interpretable are these bottlenecks? Yeah, that's a very good question. So we looked into that in an exploratory way.
Yeah.
And it definitely somehow allocates around, for example, clusters or cell types. So they are interpretable, but we didn't dive very deep into that because in the end, we couldn't get a straightforward benefit from that.
Yeah.
Yeah,
I've tried using them, and I find them a bit maybe not finicky, but like if you really want to get the most out of these bottlenecks, it could be a bit you have to dig in. But so I was curious.
Yeah.
But in addition to the advantage of simplifying the problem, it's very valuable.
Yeah, true. By the way, at the same time, like there are also new low-rank optimal transport algorithms being developed. I think last year in Europe, so 2024 in Europe, I talked to a few. I forgot where they were from, but they basically now have a low-rank approximation which is based on the synchron algorithm and not based on basically this other kind of which is also gradient descent method, which we are building upon. So like there are more and more low-rank approximations also being developed these days. So they might still improve. So this is definitely a space for future research.
And maybe also potentially like neural variants.
Yeah, I'm going to touch upon that later in the outlook.
Excellent. And just a minor comment. We don't really see your face sometimes when you're talking. So.
Am I too much on the side? All right. Perfect. Okay.
You have a good smile, so might as well capture that too. So.
Great. Okay. Thanks for letting me know.
Okay.
Cool. Then, the last point was the user experience. So maybe you might have heard about SCVers, like this organization where we try to make most single cell applications accessible, also well maintained and easily usable to users. And we wanted to bring that into one format such that we can nicely interact also with other toolboxes. For example, there's with whatever trajectory inference method you want to use from moscot, there are like different ones, the temporal problem, spatial temporal problem. I'm going to talk about that later. There's also the problem with a prior on if you have lineage barcoding readouts, you can easily interface with cell rank and do your cell rank downstream analysis, which you might know maybe in combination with RNA velocity-based methods very easily and straightforwardly. So these are the main ideas of moscot, of developing moscot. And now we can just briefly zoom into the scalability again, what it actually means in practice. Building upon these benefits of on-the-fly cost matrix computation, which turns your linear optimal transport problem from quadratic to linear, and in memory, GPU acceleration, as well as the low-rank approximation. So speaking about the performance on an atlas, we see that, for example, for 75K cells with the original Waddington OT implementation, you cannot even do that on a server, let alone on a laptop, whereas you could even run moscot on your laptop if you wanted to up to 300,000 cells. If you have a GPU, of course, that's much faster, but also in terms of runtime, So here these runtimes are given in, obviously run on GPUs. So Waddington OT doesn't run beyond 75K cells if you have limited or like not very, very huge amounts on your server of memory, whereas it's easily manageable with moscot, and you get even for 300,000 cells down to 15 minutes, what is basically what Waddington OT requires for, I think, 20,000 cells. So it's just very scalable.
Do you find that these approximations lose any signal that you expect to see that's relevant, or is there something that you'd really be missing?
Yeah, so there are definitely limitations of using low-rank approximations. The main one being that it's got more hyperparameters, so you need to select a step size, which can be crucial. And if you don't, so normally we also say how to, we provide suggestions about how to use that, but I wouldn't blindly trust it. Whereas, for example, with a full rank linear optimal transport problem, if it converges, you know it perfectly converged because it's convex. So this is, for example, one problem. For the linear optimal transport problem, it renders the problem non-convex. For fused Gromov-Wasserstein or Gromov-Wasserstein, which we're going to talk about later, sorry for mentioning this again already. I'm going to introduce it later conceptually again, but this is already not convex, although if you choose epsilon large enough, there's a recent paper showing that it actually almost renders it convex. Whereas, the low rank approximation is definitely less convex than that. So there are definitely limitations, but we still found, and I mean, this is also a contribution of this work, we still found it to be meaningful. At the same time, it's also sometimes not really easy to disentangle what effect is, let's say, if we have some sort of feature which we dislike about our optimal transport map, which cannot be explained from a biological point of view. Is it due to the algorithm not converging well, or is it due to the OT assumption to actually applying optimal transport to that problem? Is that the problem? So this is always something which is very hard to disentangle. But in terms of, if we just talk about the quality of the mapping, we have a use case where we compare the full rank versus low rank optimal transport on mouse embryogenesis, and we found it to be almost equally performant there.
Wonderful answer. Thank you.
Great. Then there are 4 use cases I would like to walk us through. First, we are going to start with trajectory inference for multi-omic data. So this will still be just the classic, let's say, Waddington OT use case with a few adaptations, for example, changing the cost function, etc. Then we're going to talk about 2 spatial applications, mapping dissociated cells to their spatial organization and aligning spatial transcriptomic slides. And finally, we introduced a novel problem in moscot, which is the spatial temporal trajectory inference algorithm, which we propose as an easy or a straightforward extension of previously mentioned algorithms, which we merge such that we now can perform trajectory inference with a prior based on the spatial coordinates. Let's start with the Pancreas use case. So we got new samples of multi-ohm, so paired RNA and attack for 3 time points. Some people might know a similar data set, which was, I think, mainly became famous in the field of computational single-cell biology with SCVelo. So SCVelo is RNA velocity method. They also used it on the developing pancreas, although the difference is that they only looked at 1 time point, plus that was purely gene expression readout. Again, here the difference is we now look at 3 different time points, and we have paired gene expression and attack. Then we computed a coupling based on a multi-VI embedding, which is from the SCVI libraries, the VAE libraries, or VAE applications for single-cell genomics, which allows to encode paired gene expression and attack data. So we computed that embedding, and based on this, we computed a graph, which allowed us to perform something which is called geodesic sinkhorn, or we basically compute the optimal transport coupling based on a cost, which is not the Euclidean cost, but is a geodesic cost, which is approximated by a heat diffusion on a graph. This is necessary because otherwise we would have got transitions which are biologically not meaningful. But we found this cost to be biologically meaningful. By the way, it's not maybe necessarily surprising that just the Euclidean cost doesn't satisfy all these assumptions, because we know that single cell data, even if it's embedded in in some SCVI embedding, doesn't really preserve doesn't preserve the Euclidean geometry or doesn't allow for all the features of Euclidean geometry. Hence, we approximated basically this manifold using an underlying graph. And then we look at these transitions from cell type to cell type. So on top, on the right, you can basically see an aggregated transport matrix. So basically, you just aggregate this coupling, which gives you cell-wise pairs or stochastic mappings on a cell mapping to the cell type level. And you would see that these transitions, which we get here, which are not dark, let's say, they are non-zero, and these you can look at and see whether they might make sense. And even if you're not a pancreas expert, you can maybe just now trust me that FEV positive is the progenitor population of the corresponding hormone-producing cell type. For example, beta cells, which produce insulin. FEV positive beta is a progenitor population of beta cells. Similarly, with FEV positive alpha is a progenitor population of alpha cells. And this is prior knowledge which we had, especially about the alpha and beta cells, whereas with the delta and epsilon cells, their ancestry is not very, very well studied. And hence we looked at that in a little bit more detail, and then looked at this part of the pancreas, which is highlighted below. So especially at those FEV positive delta 0 and 1 cells, as well as those epsilon progenitors, how they would actually evolve into delta cells or epsilon cells or alpha cells or beta cells. So what is their relationship? And this was a particular focus. Is there a question? Just because I just heard something pop up.
Yes, like a question regarding the loss of meaningfulness in the method. How do you define that here precisely? Is it just when you fail to map completely, or where you find disconnects in the trajectory that you would otherwise expect? What's the threshold for this?
So whether I basically say that this mapping is meaningful?
Yes.
Okay, right. So often the simplest way to get a first idea of whether the optimal transport coupling is meaningful is looking at that aggregated view to cell type level. And now we can let's have a look again at that transition matrix to the top right of that slide, where we can look at the mature acinar cells in E14.5. We can look at where are they mapped to in 15.5. And we see that everything is dark blue or dark violet, except for the very first entry, the diagonal entry, which means mature acinar cells will only be mapped to mature acinar cells. Right. Let's go to the second row, immature acinar cells. Where do we see something which is non-zero? This is mainly the diagonal again, so immature acinar cells, but this is fine. So basically, immature acinar cells can remain in the cluster of immature acinar cells, but we also have a non-zero diagonal element, which is the mature acinar cells. Hence, immature acinar cells will also map to mature acinar cells, which is biologically meaningful. So a little bit briefly about the pancreas. So you've got like these different branches of acinar cells, ductal cells, and then the endocrine cells. And endocrine cells are the hormone-producing cells, which we are focusing on with the alpha, beta, delta, and epsilon cells. But this was not only the example about the well-understood acinar lineage, so that immature acinar cells won't become any, for example, ductal cells or NGN3 low or high cells, but they would only evolve into acinar cells if they change their identity. And now we can also consider, for example, terminal cell states. Let's have a look at alpha cells and beta cells. We know that alpha cells and beta cells, for the most part, they won't evolve into any other cell type because they are differentiated cells. They're fully differentiated. And then we can look at the transition matrix and ensure that these cells are only mapped to themselves. So alpha cells in 14.5 should only become alpha cells in 15.5, because otherwise it's biologically not meaningful. So it's best to validate these couplings with prior biological knowledge. And for example, when you try that with the with the Euclidean cost instead of the geodesic cost, and it's somewhere in a supplementary figure in the paper as well, what you would do is you would map ductal cells directly to delta cells. And this is biologically not meaningful because we know that it's not possible that ductal cells immediately basically transition into delta cells. But it's explainable because both the ductal cells and delta cells express very highly the transcription factor HHEX. And hence the embedding is so much dominated by HHEX such that the optimal transport coupling will link those two cell distributions or cells corresponding to those two cellular distributions, because they're very close in space, and hence it's basically from an optimal transport point of view very cheap to connect them. On the other hand, if you use the geodesic cost and you build a graph, the distance on the graph to go from ductal cells to delta cells will be very, very large, because they're not close enough in gene expression space such that they are among the top, like, let's say, closest 50 nearest neighbors. But they're still sufficiently far away such that the graph is not connected between those two populations. Is that intuitive? Does that make sense?
That is absolutely clear. Thank you very much.
Yeah, I have a follow-up question. Do you hear me well?
Yeah.
So your geodesic cost is computed on your embedding, right, of the
Correct.
VA.
Correct.
So I guess that also influences how 2-point are close or not.
This is also true. Absolutely. Yes.
So why do you use a VIE and therefore are forcing the latent space to be somehow Gaussian rather than just a simple autoencoder?
Okay, so why not a simple autoencoder? This is mainly due to a standard single cell pipeline, I would say, because autoencoders are actually rarely used in single cell genomics.
I know.
This is maybe a different topic. I'm actually a huge fan for a different reason of autoencoders. And I do think there are very many limitations of those variational autoencoders. One reason is they oversmooth to a large extent. First, you always somehow build upon prior work. And then if this is a standard procedure, you just also say, okay, we accept that. We don't want to reinvent the wheel, sort of. We don't want to do every single analysis again from scratch. Second, we also have all of those couplings computed, for example, in the concatenation of PCA space in gene expressions, or PCA of gene expression space, and LSI, so this classic dimensionality reduction of attack data. We just concatenate those two spaces, and based on that, compute the K-inverse natal graph. And there basically we observe the same behavior. If we used the L2 distance, so the Euclidean distance, we would have this wrong transition, whereas if we use the geodesic cost, we get the correct transitions. So we have a baseline, if you're very interested in that, in some supplementary figure also in the moscot paper.
Okay. And the VIE that you train has a, do you force the embedding, the latent representation to also have some specific manifold, not just the Gaussian, but like to preserve the original manifold in the latent space? I know that there are some methods that are developed, like geodesic VIE or something else.
Yeah, that's also a very good point. We don't do that. So we just take the very standard pipeline of using the multi-VI implemented in SDVI with its standard parameters. Also, because this paper is really, like, let's say, an applied paper, and we really want to make sure in order to run moscot and get biologically meaningful results, you don't have to really tune your pipeline with a very certain embedding whatsoever. At the same time, it's a very, very good point. And yeah, there's a lot of work which does some geometry preserving embeddings, for example, also from our lab now at ICML. There's one about a VAE which enforces global Euclidean geometry such that you would not necessarily have to use the geodesic cost here anymore. So yeah, long story short, it matters a lot which cost or how you define your cost, and you can treat both the cost function itself as well as the embedding space.
Okay, thank you.
Cool. So we looked at the epsilon and delta cells because they were more poorly understood. And we can, for example, here to the left and visualize the epsilon cell population at the latest time point and predict the ancestors at time point 15.5 and 14.5. Here we highlight especially with this fate embedding this trajectory of epsilon cells, and we can actually now see where they are hypothesized, or where Moscot predicts them to originate from. What we then do is we correlate these pullback distributions with gene expression values of transcription factors, such that we can generate hypotheses which transcription factors could actually be responsible for the fate decision of this lineage, in that case of epsilon cells. And we actually found, we did that for all cell types, obviously, but we focused on epsilon and delta cells. And we found NeuroD2 to be the second most significant transcription factor in both epsilon and delta cells. And hence, we thought, okay, NeuroD2 somehow has to be responsible for either like one of them or both of them, or yeah, let's see what the outcome is if we do an experiment of a human iPSC cell line where we knock out NeuroD2. And we found this to have no statistically significant effect on any other cell type, including the delta cells, but it did have a significant effect on epsilon cells. So when knocking out NeuroD2, we observed a much smaller proportion of, or like a smaller number of ghrelin-producing cells, which means basically epsilon cells. And this way we found NeuroD2 to be the first epsilon cell-specific transcription factor in the pancreas. A question popped up.
Yes, but just a technical question. So, so you first start with your late time point cells, your target cell. You compute the OT to find out what are the ancestors. And then how do you identify the main factors for this transition?
Yeah, so basically if you pull back your distribution of cells at time point 16.5 back to 15.5, you get a probability distribution over your set of cells.
Yes.
And you then just correlate that with gene expression values. And basically, the intuition is a transcription factor will be active in a slightly earlier stage of the cells. And hence, we want to find the set of cells which correlates with ancestors of epsilon cells.
Okay.
So this is assuming transcription factor expression is just fixed towards the end of a developmental trajectory.
No, it's more like it precedes the actual hormone-producing cells. So if you basically look at a certain lineage, the gene expression of the transcription factor, which is highly, okay, let's put it the other way around maybe. If the gene expression of a certain transcription factor is highly expressed in an ancestor population, the corresponding descendant population is very likely to be affected by that transcription factor.
I see. Okay.
You can also do it the other way around, by the way, if you correlate it with the push forward. If you correlate the, let's say, gene expression profiles of any gene with a push forward of a transcription factor activity, you would identify target genes. And we did that in the spatial temporal problem. That way, we basically found target genes, and which were also reported in the literature, target genes of certain transcription factors, whatever transcription factor you're interested in. So it goes both ways.
Wonderful. Thank you.
Thank you.
Cool. Nice. Now we covered the maybe a little bit simpler scenario of linear optimal transport. Why is it called linear optimal transport? Because both cellular distributions live in the same space. So we're concerned with that case, for example, in trajectory inference and perturbation modeling, because both times our cells live in the same space, both times, for example, in gene expression space or in the same VAE space. But often we face the issue that two distributions, two sets of cells, live in different spaces. For example, for modality translation, you have one set of cells in a tag space and the other one in gene expression space. Or many other use cases, especially in spatial biology, which I'm going to touch upon in a little bit. So now we're looking at this formulation where we don't have the setting that both of these point clouds live in the same space as on the left-hand side, where you directly compute the distances of data points in your source to data points in your target distribution. In contrast, we now look at the middle column, the Gromov-Wasserstein optimal transport formulation, where you have two sets of points which live in different spaces. So here you just cannot take one data point from space 1 and one data point from space 2 and compute the distance between these two spaces are just so they're different. You cannot define a distance matrix on them. So what you have to do is you have to look at the relative positions of all data points within the corresponding set of cells. So you have now two cost functions. Let's look at the example of gene expression. It's basically your source distribution in red is in gene expression space, and your target distribution in blue is in attack space. Now you have to compare all the costs in gene expression space with all the costs in attack space, and that way link it. I'm going to make an example later. And then there's the third way, where we have now a mixture of the Wasserstein optimal transport formulation and the Gromov-Wasserstein optimal transport formulation, which is called hence fused Gromov-Wasserstein optimal transport. And there you have partially comparable spaces, which means if you're given a data point, let's say the first n coordinates share the same space with the target space, but the remaining m coordinates live in a different space than the remaining L coordinates in the target space. One example will be, for example, when we want to realign spatial slides, because the spatial coordinate systems, well, they would be in theory comparable, but we say they're not comparable because they might have been warped, they might have been rotated. Hence, you don't want to take them as absolute, and hence you cannot take distances of data points in the first coordinate space to distances in the second coordinate space. Whereas gene expression would be still in the same space if you have the same target panel in your spatial transcriptomics experiment. So more intuitively, what does that mean? We look at the Gromov-Wasserstein case, so if not the fused one, because that is even more complicated to visualize. But let's have a look at the Gromov-Wasserstein problem, where we now have our source distribution in red again and our target distribution in blue. And you want to link, in that case, x1 to y3 and x2 to y1, because you want to see that their relative, or you want to check if their relative positions within the distributions are similar. And here you can see, for example, x1 to x2 is already similar to y3 to y1. Now you have to iterate through all of these combinations and check how similar they are. Oops, sorry, that was too fast. So now if we assume that, or like visually also already see that x1 to x4 might be relatively equidistant to y3 and y5, that would be another good indication that x1 should be mapped to y3. So you really look at the relative positions, you iterate through all of the pairwise distances in each the source and the target distribution, which already, you see while explaining it, makes it much more complicated, and there are many more computations to be performed. And hence, this is where this computational complexity, this cubic computational complexity comes from. But it is possible to solve, again, in practice with the entropic regularization, and can be solved with also gradient descent-based methods, which makes use of the synchron iteration also in the inner loop. So there we have just basically an outer loop and an inner loop. It's a little bit more complicated to solve. It's not convex, unfortunately, as I mentioned before. Again, if epsilon is sufficiently large, it is almost convex. So this is also nice. But again, that's the trade-off between fuzziness and basically signal in the map. In the end, the output will be the same though. We will get a coupling matrix, stochastic coupling matrix which links cells in the source distribution to cells in the target distribution. Yeah. Now let's have a look at applications thereof. I didn't now do a toy example for fused chromoplastic, but the idea of fused chromoplastic, as I just said, is that you have an additional shared space, and this will encourage your mapping to go also the way such that the linear part of the optimal transport coupling is meaningful. And this fused chromoplastic case is actually, or formulation is the use case we have in most moscot problems, because you often have some shared space and some non-shared space. So for example, here we look at the task of mapping dissociated cells to their spatial organization, which was initially introduced by Mornitz-Zahn, also published in Nature in 2019. We make it multimodal and especially now scalable, because as I said before, that was cubic in time and quadratic in memory, which just prevents it from actually being applied to proper spatial transcriptomics datasets, which are normally rather large. So what do we do here? We have dissociated site-seq data, hence we have gene expression and proteins. On the other hand, we have a reference data set from the spatial liver, where we measure a subset of genes, because we have to choose the gene panel that we have on top of the spatial organization. So our coupling is informed by all of these components of the shared gene panel, but also of the non-shared genes and the protein abundance on the site-seq part. And on the spatial coordinates, you can incorporate them into the coupling, which basically encourages proximity in spatial coordinates to be mapped to proximity in gene expression space and protein space. So the other way around, we assume that in general, cells which are similar in gene expression and protein space are also rather close by in space. And that now helps us for mapping markers which were not in the panel of the spatial transcriptomics datasets, but which are key to identify portal veins and central veins, as well as identifying endothelial cells and Kupffer cells. And this way we can enrich the view of our data and have a more meaningful annotation. Now, a different use case was originally proposed by Zayed et al. In 2022. Again, not scalable. So we tried, or like basically we made it scalable using the slow rank approximation of fused chromoplastic, where we have 3 different slices of the mouse brain. And you can already see to the left-hand side, they have been rotated and they have been warped to a certain extent. And now we want to realign them to get like a more profound statistical analysis, for example, in terms of neighborhood enrichment. Now, how do we compute the coupling? We have this shared space because we have the same gene panel for all 3 slices. And we have these incomparable components, which are the spatial components. So again, maybe here it's very clear, you just cannot take the distance with respect to the spatial coordinates in the same space, because these are different coordinate systems which are not comparable. So you can only compare the relative position within each single brain slice. And then we have 2 downstream methods how we can align them. Either we do an affine alignment, which means we only want to linearly translate or rotate the slices. So what is highlighted here, you can see on the top right of each slide, is that the specificities of each of these slices are being preserved. For example, on the blue one, we have like a small hole, whereas in the middle one, we have a dent. On the right one, there's like no such specificity. It looks just like a very nicely formed brain. And with the affine alignment, because we only rotated it and linearly transformed it, we basically keep these features. Whereas if we use warping, we really enforce these brains to nicely align. So because here we chose the blue brain slide to be the reference, we now also remove the dent and introduce that hole basically in the orange one in the middle. And the green one, we also made this hole now to align really nicely with the reference slide in blue. Which one you want to use of those two methods, I think, is really a question of the biology, of how similar they are. Depends on whether you just roughly want to align them or whether you really want to force them into the exact same coordinate system. But this is how, moscot can be used for aligning large-scale spatial slides. And now the last application would be this spatial temporal trajectory inference method, which we introduced. So basically now we have spatial transcriptomic measurements across time points. And we always profile genes as well as spatial coordinates. So I think that's a stereo-seq panel, or stereo-seq technology. And again, the optimal transport coupling is based on the same information as the alignment. So we compare gene expression in the same space and the spatial coordinates in incomparable spaces. There are a few details which make it different, though, from the alignment problem. I'm not going to go too much into detail, but if you have development, you can have a prior guess based on proliferation and apoptosis markers, which cells might proliferate more or die, which happens obviously a lot in development, and hence there's a few details which are different than in the alignment problem, but in the end, the information based on which we compute the coupling is the same as in the alignment problem. So what we can do with that is then, for example, track whole organs across time. So on top we see the original annotation, and then we map it to see where we can actually locate or where we predict the heart to locate in future time steps. And then you might wonder, okay, do we actually need it? Why couldn't we have just applied the normal Waddington OT use case, the normal temporal problem of moscot? And hence we did a benchmark, or we compared it. We developed a score, which basically says a transition is correct if biologically it makes sense, such that we map on a cell type level the precursors to descendants, whereas we would penalize such a mapping, as I just outlined before in the Pancreas example, if it maps across lineages, where we know the lineage tree basically doesn't allow you to go downstream and still reach that cell type. And based on the score, we compare the spatial temporal problem with the temporal problem, and also to TOME, which is a KNN-based approach for trajectory inference. And we see that spatial temporal actually is beneficial compared to not incorporating the spatial information. Right, so all in all, moscot unifies optimal transport applications and single-cell genomics. We've got these different solvers implemented. I didn't mention all of them. I only basically walked us through the ones which we defined or which we incorporated into the moscot storyline and the paper. But as a brief overview in trajectory inference, we have the temporal problem, which we looked at in the mouse Pancreas example, the spatial temporal problem, which we just looked at with the developing mouse embryo, the lineage problem, which incorporates as a prior the information you get from barcoding experiments. In the spatial transcriptomics domains, you can have a mapping problem for mapping dissociated cells to their spatial organization. There's also the alignment problem for aligning spatial transcriptomic slides. For multi-omics, or for translating between different modalities, we implemented the translation problem, which is based on Scott, which is the method which originally proposed something along the lines. And then we also offer interfaces, nice undata interfaces with very easy to handle downstream methods like these cell type aggregations. And we basically call them according to the underlying optimal transport problem, whether they solve a synchron gramm-of-assistant or fused gramm-of-assistant problem. And we have a few notebooks, for example, also on them, how you can use them for analyzing your perturbation data as well. So I just encourage you to check out moscot.tools, although I was told that this website is down at the moment, so just go to moscot.readthedocs.io for more tutorials and examples, and reach out if you've got any questions, and feel free to contribute anytime. Cool. Before giving an outlook, maybe a brief break here whenever if we've got a few questions about the discrete moscot implementation.
Yeah, I was about to say, we're almost right on time. What do you have? Well, first of all, let's thank our speaker for the wonderful presentation before we actually do that. And yeah, we're right on time. Would you have a question, or sorry, time for a few questions from the audience?
Sure. Yeah, I can run over time. I don't have anything else afterwards.
Wonderful, absolutely. So let's open that. That opens up our section or question section for the audience. So if anybody has any pressing questions about the method or anything in the presentation, please do speak up now.
Yeah, I think Joao might have.
Yeah, Joao?
Yeah, I had a question just because, like, you actually mentioned this just before in the previous slide. Just how are you doing to make the optimal transport given that the amount of cells changes right through time on those kind of measurements? Like, do you just consider the initial cells and you're just looking at their trajectories, or you put some kind of weight on like if this cell is going to proliferate more? Because this is something I've run with like in database, which I don't know exactly the best way to solve. I was just wondering what you did here.
Yeah, so first, it doesn't matter how many cells there are in each population. So it doesn't matter whether you map 10 to 100, 10 to 1,000, or 10 to 10,000. So the structure, let's say, will remain the same of the optimal transport coupling. You will still map the same cell type to the same cell type. So the actual absolute number doesn't matter. What does matter is whether you assign a certain cell a higher prior weight, basically. So if you think about it in a mathematical way, actually what we look at is a mixture of Dirac distributions. And then you would just pose some weight on a certain data point. So you multiply that Dirac instance by a larger number such that a single cell which is assigned a higher weight, will develop into a larger number of cells than the cell for which you didn't increase the weight. So this is why you have these marginal distributions. And by default, you just assume all the marginal or all the cells have equal weight. So basically you have a uniform distribution of the cells. But if you have prior knowledge about proliferation, you can just increase the weight of a single cell in the source population, because you will know, or you have a good reason to believe that this cell will actually differentiate into two different cells. So it has two descending cells. Whereas if it expresses many apoptosis genes, you can already decrease the weight of that single cell in the source population because you actually don't want to map it. So you just say, okay, let it die. So actually, I don't want to map it. Now, if you don't know the score beforehand, what you can do is you can do something which is called unbalanced optimal transport. In moscot, you steer these values with tau A and tau B, and let's say there's epsilon as an important parameter, but then there's definitely also tau A and tau B if you want to use it, which are good to be understood. So that basically tells you to what extent you allow the algorithm to discard cells automatically or to increase the weight or the mass of single cells automatically. So from a mathematical point of view, if you discard the mass of a cell, or if you discard a cell, this obviously makes it cheaper to transport your distribution because it's just fewer cells which you have to transport, right? So this doesn't make sense to just allow for discarding, because in the end we would discard everything and then the total cost would be zero, basically, of transporting our masses. So this is why you normally have you derive it by having a loss of your prior mass and the one which the posterior marginals, basically. So you want to allow a certain deviation of the mass, which is computed with the KL divergence between your prior mass and your posterior mass, what the algorithm assigns, basically. So you've got these two different parameters. Either you can yourself assign or score the genes for proliferation and apoptosis, and based on that, adapt your marginal distributions. We also have a function implemented in moscot, so you can just use it. There are tutorials on that. And the second thing is you can also just, from an optimal transport point of view, perform unbalanced optimal transport. And mostly a mixture thereof is best.
All right, thank you very much. It's clear.
Do we have any more questions from the audience? Continue, please.
Yeah. Hi, Dominik. I want to ask some question, but not about the moscot paper, but the cell flow, like the recent paper of yours.
Okay.
How do you think of because you mentioned you would introduce some later. Yeah, please.
I mean, I don't know how much time we have. Yeah, maybe the cell flow is most interesting then. So maybe just as an overview, very, very briefly. So this is all discrete optimal transport. There's also the concept of neural optimal transport. So we generate basically, especially if, for example, the case of images is very natural that we might not have the perfect match. So we generate our match. This is based on an ICLR paper from last year. And then we are still missing a few things to make all the moscot applications neural. So we have to also formulate the chromafascistine and fuse chromafascistine formulation, which is the nearest paper from last year, but I don't want to talk about that because in the end it culminated into cell flow, which allows you to perform many different tasks, but you can think about it as a toolbox for perturbation modeling. And we have a few different use cases, for example, to predict donor-specific cytokine treatment responses, to model the perturbed development of entire embryos, to do these classic cell line tasks of modeling the transcriptomic response of unseen molecular and genetic perturbations. And then we also have two use cases for protocol optimization or cell fate engineering. So basically we look at which protocol we should use such that we obtain certain neurons, or also in the case of brain organoids, it's very, very complicated protocol. So you have in that screen up to 23 modulators actually, and in the original training data set, you can have combinations of up to 10 different modulators. And then you have a continuous timing is basically when you expose the cells to these morphogens, and hence you open up a huge space of protocols. And what we then do is performing a virtual screen, so that you can say, okay, you don't have to do all of these 23,000 protocols, which we selected as a subset of the potential space yourself, but we just generate everything in silico. So this is based on flow matching mainly. So yeah, maybe a brief outlook to future work, which might be interesting.
Yeah, just wondering, how do you think of for generative modeling for single cells? It's a very general question, but yeah, just want to ask what's your opinion on this topic, because I think the most biggest concern is the generated states or cells, they cannot be evaluated, right? There is no ground truth. So how do you think of this generative modeling?
Well, I do think you can evaluate that properly because you do have these measurements, right? So you can just leave out cells corresponding to a certain treatment, for example, during training. And this is how you evaluate these models, right? And then you generate the cells, and then you compare the generated cells with the true cells. And because it's on a population level, you just compute the distributional distance. So this is why I think this is the most meaningful distance. You compute the distributional distance between true and generated cells. So what exactly do you mean? Maybe I misunderstood your question. You cannot really assess these models?
What I mean is the only thing we can do is to evaluate the population of the cells, but not the single cell. For each individual generated cell, we cannot say it's not like proteins. Like protein, they have very much more stable and statistic structures, so people can evaluate with biological methods. But for cells, if you destroy one cell, then it's done. And if you generate one cell, you don't know how to use biomedical methods to create one cell, right?
But if you consider that from a statistical point of view, then you could argue that if you observe all of the cellular states corresponding to a certain condition, hence you want to have like a large sample size of your population, this should be representative or should reflect the true distribution of cells corresponding to that treatment.
Okay.
Of course, this is not on a single cell level, but in the end, I think you're more interested in what a treatment does to a population. How does the population change in general? Don't you think so?
Yeah, that makes sense. Okay, maybe let me change the question. Like, what do you think is the biggest challenge for this area? Like to, for example, your work cell flow. Like, I think it's very important. It's model very properly and to the perturbation data. So what's the next challenge you think you are going to do, or you think that's very important for this direction?
Okay, yeah. So there are quite a few limitations. Limitation one, which we face in the course of that work, is that those flow models mainly perform well when being trained in some sort of latent or denoised space. Now, the problem is that VAEs in single-cell genomics are not particularly good at generalizing and decoding, hence decoding generated cells. So even if the generative process might be good, if you generate a very, very good cell in a latent VAE space, you still have to reconstruct them. Hence, you get an additional error through reconstruction, which is not due to the generative process of your cell.
Yeah.
Which is, by the way, why I mentioned before that I like autoencoders, because autoencoders are much better than variational autoencoders for that task. But yeah, this is, for example, one very open question. Another one would be that we found in the course of CellFlow that predicting completely unseen treatments, where you're only based on the embedding of your, for example, genetic knockout or of your molecule, there CellFlow might be better than competing methods, but I don't think it's very useful, to be honest, to biologists, because it's still not good enough. So I think we need to find better ways how to represent these interventions. And for example, making use of functional embeddings of drugs or genetic knockouts rather than purely structural ones, because in CellFlow we only use structural embeddings of these interventions.
Yeah, okay, thanks. That's very insightful.
Thank you.
Wonderful. So in the interest of time, let's thank our speaker one more time. Dominik, that was an amazing presentation. Thank you for agreeing to present in the multi-omics reading group. And that concludes the 18th edition of the reading group. Thank you very much to everybody who attended.